Hi, Assalamualaikum. My name is Saidah, and today I will be presenting my final year project entitled B-cell epitope prediction of dengue virus NS1 protein using bioinformatics tools. The B-cell epitope is a portion of antigen or the antigenic determinant that can be recognized by the B-cell lymphocyte to elicit adaptive humoral immune response. So in my research paper, I mentioned a little bit about the epidemiology, pathology, and the genetic information, especially on the NS1 protein of the dengue virus. I also mentioned about the bioinformatics tools to predict the linear B-cell epitope, which include BPPRAT 2.0, BCPRAT, and SVM3. The B-cell lymphocyte has a membrane-bound immunoglobulin or antibody. This antibody has a paratope region that can recognize the epitope or the portion of the antigen. So the binding of the paratope to the epitope region of the antigen can elicit adaptive humoral immune response. The B-cell epitope can be present in either linear or continuous structure or in conformational or in discontinuous structure. But in this research, I'm more focused on the linear B-cell epitopes. The dengue virus has four serotypes. The dengue infections could lead to a wide range of clinical manifestations, from classical dengue fever to a severe dengue hemorrhagic fever and dengue shock syndrome. Dengue virus is categorized under family Flaviviridae and the genus Flaviviruses. This is an envelope virus with icosahedral symmetric capsid and positive sense single-stranded RNA. Its genome has 11,000 bases approximately and can encode single large polyprotein. The polyprotein could be subsequently cleaved into structural and non-structural protein. There are 7 non-structural proteins, but I'm only focused more on the NS1 protein. The NS1 protein is highly conserved among the dengue virus serotypes. It can aid in viral replication process and exists in 3 forms, which are monomer, a dimer, and hexamer. As this protein is highly conserved among the dengue virus, this protein could be a good candidate to create a peptide vaccine against dengue virus. Antibodies that race against secreted NS1 proteins may cause endothelial dysfunction due to cross-reactivity with host proteins and endothelial cells. As mentioned by Chen et al. In their research article, immunizing animals with full length of NS1 protein, especially by aspirated dengue virus serotype, may encourage the antibodies to cross-react with the host proteins, resulting in deleterious consequences both in vitro and in mice. Therefore, to avoid cross-reaction of the antibody, we need to predict the epitopes region of the NS1 protein instead of using the whole NS1 protein sequence to create vaccine. The epitopes of the NS1 protein can be predicted using bioinformatics tools. There are 3 linear B-cell epitope prediction tools has been used in this study, which are BPPRAT 2.0, SVM3, and BCPRAT. So the BPPRAT 2.0 can predict B-cell epitopes from the antigen sequence. It uses a random forest algorithm trained on epitopes and non-epitopes amino acids determined from crystal structure. This prediction tools has outperformed other available tools for sequence-based epitope prediction. Meanwhile, SVM3 is an analysis method that combines propensity scores and tripeptide similarity to improve prediction accuracy. The last prediction tools used is BCPRAT, which also applies SVM or support vector machine models with a subsequent kernel. BCPRAT has outperformed 11 support vector machine-based classifiers with an AUC of 0.758. The subsequent infection of different dengue virus serotype may lead to cross-immunity and worsening the disease through antibody-dependent enhancement or ADE. Nowadays, there are still no specific antiviral drug or efficient vaccine to treat dengue infection. The first licensed dengue virus vaccine was tetravalent Dengue, but this vaccine has not been approved in all countries. In addition, this vaccine only has 76% efficacy and can only be used to patients with a previously laboratory confirmed dengue disease. Injecting Dengue to people with no record of dengue virus infection might lead to severe hemorrhagic dengue fever. So to construct a vaccine, we need to predict the epitope. However, the other reliable B-cell epitopes prediction techniques, such as nuclear magnetic resonance and X crystallography, is very expensive and time-consuming. Move on to the significance of the study. First, identifying conserved epitopes can develop subunit vaccines that broadly effective for all dengue virus serotypes. Second, predicting the epitope of the NS1 protein can be the prominent methodology to develop peptide vaccine against the dengue virus. Third, epitope identification using immunoinformatic or bioinformatic tools is useful in designing peptide-based vaccines as it is much more cheaper, faster, and flexible. So overall, the objective of this research is to predict the linear B-cell epitope of dengue virus NS1 protein using BPPRAT 2.0, PCPRAT, and SBM server. Second, to analyze the conserved epitopes using the epitope conservancy analysis tool. And third is to visualize the conserved epitope sequence using WebLogo 3.0. Move on to the methodology. First, I went to the NCBI Gene Bank to collect 48 FASTA format of NS1 protein samples from 4 countries in Southeast Asia, including Malaysia, Thailand, Indonesia, and Singapore. So the samples of NS1 protein has originated from the dengue patients, mostly from their blood serum. Next, I predict the linear B-cell epitopes of the NS1 protein with this accession number, which is the dengue virus serotype 1 from Malaysia, using BPPRAT 2.0, BCPRAT, and SPM3. Next, I use epitope conservancy analysis tool to analyze the conservancy level of each predicted epitopes. Next, I use cluster W to align all the 48 FASTA format of NS1 protein samples. Then, I visualize the conserved sequence from the sequence alignment here using WebLogo 3.0. Finally, I compare the predicted epitopes with the previous study to observe which predicted epitopes regions has overlap with epitopes with proven immunodominance. Based on the result, we can see that from the whole 352 amino acid sequence of NS1 protein, there are 24 epitope region has been predicted by the 3 prediction tools. These epitopes has a size range from 6 to 25 amino acid. So the 3 prediction tools has agree well with nearly the same areas indicate as epitopes by more than one prediction tools. For instance, epitope 7, 8, 9, and 10 has overlaps with each other. The overlapping epitopes has been summarized in a table which I will show later. Move on to the conservancy analysis result. There are only 7 predicted epitopes that were found to be substantially conserved over 80% within a particular dengue virus serotype. Meanwhile, there are 11 epitopes were found more than 70% conserved amongst the 4 dengue virus serotypes, with 4 of them, which are epitope 12, 15, 22, and 23, has the highest conservancy level, which is 85%. So this table here shows the sequence of the conserved epitopes that visualized by WebLogo 3.0. This table here shows the conserved epitopes that overlapping within different prediction tools and by previous studies. From this result, we can see that epitope 12 has overlapping with the epitopes identified by Saxena et al. And Zhang et al. Meanwhile, epitope 15 has overlaps with the epitope predicted by Pushpakumar et al. So as these 2 epitopes has overlaps with the epitopes that already has proven immunodominance, therefore epitope 12 and 15 can be a promising therapeutic candidates against 4 dengue virus serotypes. Meanwhile, epitope 22 and 23 also has the highest conservancy level. However, it is located at C-terminal regions, which according to Pushpakumar et al., epitope at this region also conserve with the other 2 flaviviruses group. Therefore, if we want to create vaccines based on these epitopes, the vaccine might not be very specific towards dengue virus. Meanwhile, according to Chen et al., the antibodies that raise against the epitope at C-terminal region can cross-react with vascular epithelium. Therefore, epitope 22 and 23 should be ruled out as a treatment candidates for all dengue virus serotypes despite having a high conservancy level. To wrap up, the bioinformatics tools is basically a promising approach to predict epitopes as it is more flexible, cost-effective, and time-saving. From this research, 2 epitopes were predicted showing the highest conservancy level across all 4 dengue virus serotypes, indicating its potential as a vaccine target or biomarker for dengue virus group detection. Nonetheless, these predicted epitopes must be validated further in a laboratory before any application. Besides, predicting the linear B-cell epitopes alone is insufficient to create peptide vaccine against dengue virus. We also need data of other additional properties such as the existence of conformational B cell epitopes and T cell epitopes. Finally, we could increase the number of samples by covering samples from another region other than Southeast Asia, such as from Brazil and other countries that dengue is an endemic. That's all from me. Thank you.